one of the advantages of winning the Nobel Prize was that I became acquainted with these two gentlemen that you've both heard about, Jens Skou and Paul Boyer. I want to pay my personal tribute to both of them. They both died at more or less the same time, at more or less the same age, just before their 100th birthdays. They were both great scientists, and it was a great privilege to have known both of them. So the first question I want to address in this lecture is, where does biological energy come from? We require energy to do everything we do, to move our muscles, to think, as we've just heard about. 30% of ATPase effectively used in neurobiological processes. And the answer is that a lot of biological energy comes from the sun. And I'll explain how that energy is harvested in a moment. And another source of biological energy are thermal vents in the oceans, where the energy is derived from processes occurring in the core of the Earth. Now, we're familiar in our current world of methods of harvesting solar energy using these solar panels. And also, it is perhaps not so immediately obvious that hydroelectric power stations also harness solar energy. The sun heats the oceans evaporate, the evaporated water forms clouds, the clouds then precipitate rain in the mountains, we collect the water, and that stored water represents a form of stored potential energy, which then can be released through turbines to generate electricity. In the biological world, we generate or we harvest energy by the process of photosynthesis in green plants. This is a very complex set of chemical reactions that occur there, and a very complex organization of many proteins are required to harvest the light and to use it for this possibly the most important chemical reaction on Earth, namely the splitting of water. So this is referred to as the water splitting reaction, where solar energy splits two water molecules to generate oxygen, and here it looks like hydrogen, but effectively reducing equivalents. So these are this is the light reaction of photosynthesis. And then in the dark reaction, the reducing equivalents, the hydrogen, is combined with carbon dioxide, and this produces organic molecules, carbohydrates, for example. So this is the way that light from the sun is harvested. Now, one of the things I do is I go around schools and speak to school children. I actually recently went to my granddaughter's school, and one of the things I did was to take green leaves from my garden, and I put them in a beaker of water. I shone light on them, and actually you can see the oxygen start to evolve from the stomata in the leaves. I then collected the oxygen and put a spell into it and pop. And the children love this. I gave them all leaves, and they put them in beakers, and they watch the oxygen bubbles coming out. Now, I thought that was a fantastic demonstration. It's actually a reprise of an experiment conducted in the 18th century by Joseph Priestley, where he took sprigs of mint and he put them in bell jars where he'd remove the oxygen by burning. And he put a mouse in there. Without mint, the mouse expired immediately. When he put mint in there, the mint generated oxygen, and the mouse survived. That's a fantastic experiment, because it is one of the ways in which oxygen was discovered. It was discovered contemporaneously by Lavoisier and also by a Swede called von Scheele. But it was also the discovery of photosynthesis. What a fantastic experiment that was. Now, the way we harvest solar energy is on an industrial scale by growing crops. So we entrap the energy in crops. The crops make carbohydrates. We then consume the carbohydrates in the form of food, or we feed them to animals and then kill the animals and consume the energy in that way. In the human body, the essential organelle involved in energy conversion, in other words, for taking the energy that we've consumed in foodstuffs, is the mitochondria. So the mitochondria occur in all human cells except for the red blood cells that are circulating around us. And when I first started to work in this field in 1978, this is the way we used to think about them. So they are unusual insofar as they have a double biological membrane, an outer membrane, and then an inner membrane, which you can see is highly folded and invaginated. And these are called the cristae mitochondriales, in other words, the cristae of the mitochondria. And it's here in the cristae that the central reactions involved in energy conversion take place. Now, what the other half of the picture is showing us is that our concept of the mitochondria as being some kind of bacterial-like organelle swallowed up at some early evolutionary event is far from the truth, because actually within a cell, so this is part of a skin cell, the mitochondria are all joined up into a kind of reticulum. And you can see the reticulum is moving around and it's actually being remodeled. And bits are breaking off and other bits are fusing with it. And the mitochondria in reality is either in this highly reticulated state extending throughout the cytoplasm of the cell, or under certain conditions during cell death, for example, the mitochondria break up into little vesicles. And this, what we've learned in the last 10 years is that the mitochondria are extremely dynamic, and we don't understand the reasons for this. I just mentioned this in passing so that you have a up-to-date account of what a mitochondrion is. Now, the most important reactions occurring in mitochondria are very simple. So if this sphere represents this highly invaginated inner membrane, what is going on is that the chemical energy that we're deriving from consuming foods, the carbohydrates, the fats, that chemical energy is being used to do a very simple process, and that is simply to move hydrogen ions, protons, from the inside space to the outside space. That is where the energy is being used. And so the effect is then to build up an excess of hydrogen ions on the outside. And this excess of hydrogen ions is a source of potential energy. It's stored energy that can be coupled to other processes and be made to do work. And this process is referred to as chemiosmosis. It's an idea that was developed by a British biochemist called Peter Mitchell, who was the recipient of the Nobel Prize in Chemistry in 1978. So Mitchell talked about something called the proton motive force. That's what this black box is. It's the proton motive force. And he coined that phrase by direct analogy with electron motive force that we're more familiar with, voltage. And effectively, proton motive force in mitochondria is a voltage. Across that inner membrane of the mitochondrion, there is a voltage of minus 200 millivolts. And that voltage is being used to do work in a way that I'll explain in a moment. Now, once you've generated proton motive force, and you can do it in different ways, you can do it by the process that I've been describing so far that occurs in mitochondria, the breaking down, the oxidation of sugars and fats that we refer to as respiration. So this is the point where almost all the oxygen that we're breathing in is consumed. So the photosynthesis is splitting water and generating oxygen. In our mitochondria, we're taking that oxygen and we're converting it back to water and using that energy to generate the proton motive force. So you can generate it by photosynthesis directly, as in green plants, by respiration in the mitochondria, and by other means. And once you've generated it, you can actually use it to make heat. So newly born children have a mechanism called non-shivering thermogenesis. They have difficulty in generating and controlling their body temperature. And their mitochondria are what we call uncoupled. And they simply release the proton motive force, and this generates heat. So this is non-shivering thermogenesis. And then it can be used, I've done what I didn't, okay. And then it can be used to, for example, to make the flagella rotate in motile bacteria. So proton motive force is the source of energy to turn the flagella in motile bacteria like Escherichia coli or Salmonella typhi. But most importantly, from the point of view of this presentation, it's the source of energy to make adenosine triphosphate ATP from the breakdown products ADP and phosphate. And the next part of the talk will be about that process. So let me first describe what happens in mitochondria in quite general terms. So the sugars in our foodstuffs are broken down by a series of biochemical reactions outside the mitochondria. And so this again represents the inner membrane of the mitochondria. We don't worry about the outer membrane, it's very porous. So these processes, referred to as glycolysis, break down the sugars and generate a molecule called pyruvate. Pyruvate is then transported into the mitochondria through a transport protein. It encounters an enzyme called pyruvate dehydrogenase, which converts pyruvate to acetyl-CoA and at the same time generates reducing equivalents in the form of a molecule referred to as NADH. The acetyl-CoA then feeds into a famous biochemical cycle, sometimes called the tricarboxylic acid cycle, sometimes the Krebs cycle. And without going through the details, this cycle generates more reducing equivalents in the form of NADH, here, here, and here, and in the form of another molecule called FADH2. Now, what about the fats? So the fats in foodstuffs transfer their fatty acyl side chains to carnitine. So first of all, they're transferred onto coenzyme A, and the acyl-CoA transfers the acyl groups onto acyl-carnitine. Acyl-carnitine is then transported into the mitochondrion, and the opposite occurs. The acyl groups are transferred back onto coenzyme A inside the mitochondrion. So this is an indirect mechanism for transporting acyl-CoA through the membrane. The acyl-CoA then goes into another complex set of biochemical reactions referred to as the beta oxidation pathway. And again, without going through details, the net effect of breaking down those acyl chains from fats is to generate more reducing equivalents. So the summary of what I've said so far is that the energy from fats and sugars in foodstuffs are used in the mitochondrion to generate reducing equivalents. Those reducing equivalents then feed into a series of complex enzymes, referred to as complexes 1, 3, and 4, which use that energy from these high energy electrons, the reducing equivalents. They use that energy to simply translocate protons from the inside space to the outside. And so this is how the proton motive force is generated. Now I'll show you a little bit more details, because over the past 40 years, what we've done is to work out the atomic structures of all of the enzymes involved in this process. So this is the entry point where the reducing equivalents feed into the chain. The electrons transfer through 100 angstroms of protein here. They're transferred to a quinone, which becomes reduced. And concomitant with that, some energy is released, which then makes the protons go through the membrane in the required direction. Now, quinones are soluble in the membrane, and so they then carry the electrons to the next enzyme in the pathway, that we won't worry about these here, which is called complex 3. And again, more of this energy from the high energy electron, which is now to lower energy, is transferred through this process, enters via the reduced quinone, and then the electrons transfer to a molecule called cytochrome C in its oxidized form, and concomitant with that, more protons are ejected. Once reduced, the cytochrome C moves and carries the electron to the terminal enzyme, which is called cytochrome C oxidase. Here, two things happen. More protons are forced through the membrane to the outside, and a chemical reaction occurs. Oxygen is reduced back to water. So this is the point where the oxygen that we're breathing in is reduced back to water. And then the proton motive force, having been generated in this way, is used to do work by the machine that I've studied, the ATP synthesizing complex that I'll describe in detail in a moment. So to summarize, the energy from the foodstuffs is being fed into these complex enzymes. These complex enzymes use that energy to eject protons to the outside. And then the ATP synthesizing machine, by a mechanism I'll show you, uses that energy to do work and generate ATP inside the mitochondria. Now, in that location, ATP is not much use to us as a form of energy, so it has to be distributed throughout the cell and first of all brought to the outside to be made available as a source of energy to make our muscles work, to allow us to think, and so forth. And at the same time, in exchange for the ATP made on the outside, the same protein brings back spent fuel. So when ATP releases its energy in whatever process it's being required to do that, the products are ADP and phosphate. And so the ATP is exchanged for external ADP. The ADP is brought back, and another protein brings back the phosphate. Now, this is a huge process that's going on inside our bodies. Everybody sitting in this room today is going to make 60 kilograms of ATP inside their bodies to keep themselves alive. Now, that doesn't mean to say, of course, we've got 60 kilograms of ATP at any moment in time. It means that we're turning over ATP at that level. So this, on a global scale, this is a massive chemical reaction that's going on. Now, what we've learned over the past few years, past 30 years, is that these biological energy packages, ATP molecules, are made by machines. So that I've described the proton pumps briefly that harvest the energy. And this is equivalent to a dam of water. It's generating potential energy. And then a machine, which is actually a turbine, or equivalent to a turbine, uses the energy to generate the useful fuel ATP. And then, as I've just briefly described, a transport system then delivers the ATP to the biological cells. Now, I started to work on these turbines in, 1978. And at that time, this is all that was known. So these are pictures of the inside of mitochondria determined by electron microscopy and negative stain. And what could be seen, although the pictures were taken in 1962, not much had happened in the interim. But what could be seen was that the insides of the membranes, the inner membranes, the folded membranes, were covered in structures that people called mushrooms or lollipops. They had a head, which is about 100 angstroms in diameter, but that head was not sitting on the membrane, it was distant from the membrane. In other words, there was a stalk, and that's where the name mushrooms and lollipops came from. So what I set about doing was defining this lollipop and mushroom in chemical and structural terms, and that took about 25 years. The outcome was this model, because in reality it's a rotary machine, and the proton motive force is being used to generate a rotation of the central part of the lollipop, and that rotation is carrying the energy from the proton motive force into this part of the structure, the head of the lollipop. And this is where ATP is being made. Now, there are 3 catalytic sites in this head, and every time that the rotor has gone through 360 degrees, you generated 3 ATP molecules. This shows you the rotor so that you can see the synthetic part in shadow in the background. So the The rotor is being turned in the direction shown. It's actually going round at about 200 per second. So your bodies are full of these little machines rotating at 200 per second. And this machine is spewing out huge amounts of ATP, as I've described. And of course, what we've wanted to do, to understand, and we do understand it, is the chemistry of what is going on here. I'm not going to show you the details of that. And then in recent years, we've learnt about, in molecular terms, how is rotation generated? How does proton motive force generate this mechanical rotation of 200 per second by the protons, which are represented by the red balls going through the membrane domain? I won't describe that today in detail. But these rotary machines have machine-like elements that we can recognize. There's a spring that I'll show you where energy is stored transiently during the rotation. It maybe wasn't so evident, but actually the rotation of the part at the top was stepping. It actually steps through 120 degree steps, whereas the ring is rotating continuously. And this implies that energy is being stored somewhere in a spring and released in a quantized fashion to carry out the chemistry. So there's a spring, and then there's a crank in the middle, which is modulating the properties of the catalytic sites, allowing them to bind ATP and phosphate, then bring the ADP and phosphate together to bring ATP, and then to release the ATP from the structure. And part of the rotary mechanism is there's a bearing. So let me just show you some of these features. So here, this is the rotary part in the middle here, the ring in the membrane, and then this thing here, which is like a crankshaft. Here is the bearing at the top, where the top is spinning around. And somewhere in the middle, we're still not quite sure where, in the middle of this complex, there is some kind of spring-like element where energy is stored transiently. And then in the membrane part here, torque is being generated from proton motive force to make the thing rotate. Now, I don't want to go into the details too much, but if this is a depiction of the rotary part, the ring in the membrane, and it's made of proteins called C subunits. And I think in this depiction, I've drawn it with 12. And next to it is this protein here in green, which has a half channel in it. And through that half channel, the protons access negatively charged carboxyl groups in this site here. Once those negatively charged carboxyl groups are neutralized, they are then more hydrophobic. And so they move from this site to a more hydrophobic environment in contact with the phospholipid bilayer, simply by Brownian motion. This then brings the next negative charge to this site. Another proton goes in, neutralizes the carboxyl. This then moves to this site. So you can see that successively, as more protons go in, they get carried around the ring until they reach this site here, which has chemical properties that makes that neutralized carboxyl reionize, regenerate the negative charge, and release the proton on the other side of the membrane. That was a postulate put forward by German scientists called Wolfgang Junger, and the structural work that we and other people have done in the last 5 years has shown that this mechanism is almost certainly correct. So the important point to remember is that if there are 12 of these C proteins and each of them carrying a negative charge which becomes neutralized, if there are 12 in the ring, to generate 360 degrees of rotation, you would take up 12 protons. So a 360 degree rotation would consume 12 protons. And remember what I told you a few moments ago, every time the ring goes around in the part not shown, the catalytic part, you've generated 3 ATP molecules. So the cost of making each ATP is translocation of 4 protons. So I'll just summarize that. So each 360 degree rotation generates 3 ATPs. A full rotation requires the translocation of 1 proton per C protein. And therefore, the proton to ATP ratio, which you can measure experimentally actually, is the ring symmetry divided by 3. Now, one of the most surprising things that we found was that there are different ring symmetries in different organisms, and that the ring symmetries differ between multicellular and unicellular life. People anticipated that proton ATP ratio would be a universal number. This turns out to be wrong. So what we have shown is that throughout metazoans, that is, say, us down to sponges, the rings have 8 C proteins in them, and therefore the cost of making each ATP molecule is 8 divided by 3, 2.8. Whereas other organisms, and here it's a range of these C rings, so this is our C ring, the C8 ring. These are a range of other rings that have been found in bacteria and in chloroplasts. So chloroplast is a C14 ring. This is a cyanobacterium. This is another microorganism. These are yeasts and fungi. So all of these different kinds of organisms are paying more protons than we pay to make an ATP molecule. Now, there isn't a clear explanation of this, but if you think about these rings in mechanical terms as gears, actually what we have is the highest gear, the smallest ring, and to drive that high gear, you require a high proton motive force, a high voltage. And so the idea is that these organisms which go to the trouble of making a much bigger ring and pay more protons to generate the rotation of the ring, they're not able to drive a small ring because they've got a low proton motive force, lower. And so they have to opt for this strategy, and they pay the cost of doing so. So that's the idea. It's still not been properly tested. Now, I want to change gear myself at this point and just talk about more general aspects of bioenergetics, where I think there are really significant challenges for mankind for the present and the future. And some of them arise from our aging and growing population. And of course, one of the consequences of the aging population is an increased incidence of neurodegenerative diseases. Another thing which is extremely disturbing that's going on in the world at the moment is that infectious diseases are making a comeback. And this is because the antibiotics that we've used since 1943 in clinical medicine are becoming useless in treating infectious diseases. So diseases that we thought that we conquered in the 1940s, for example, tuberculosis, are now becoming major scourges of mankind. Unless we do something about it, we're going to be in huge trouble. So there's been an upsurge in tuberculosis. And then we have these other issues of there being an increase, an ever-increasing demand for energy to sustain our civilizations and the lifestyles that we've grown accustomed to. And concomitant with that is the related issue of global warming. So I'll touch on each of these issues. So in relation to so let me deal with this first issue. I've not said this, but one of the features of the mitochondrion is that it actually has its own DNA molecule inside it. It's a very small DNA molecule. Most of the DNA in our cells, almost all of it, is in the nucleus. A tiny bit of it is inside the mitochondrion, only 16,000 bases of DNA. And that is actually encoding for components of those complex machines that I've shown you. Not all of the components, some of them. And the interesting thing about this DNA molecule is that it derived entirely from our mothers. There is no component from our father's sperm, the mitochondria in our father's sperm, that survives in our mitochondria. So they're maternally derived. And this means that any diseases that are associated with defects in that particular genome, the mitochondrial genome, are maternally inherited. And we used to think of them as rare diseases, but actually there's been a survey done in Britain through Newcastle University, and the incidence is about 1 in 6,000 of live births. So it's still a minor disease, but a significant one. And often these mutations lead to defects, so they're going to cause defects in the energy converting process, and this manifests itself in defects in muscle and also in neurological tissues. And one of the most frequent symptoms is that of ptosis, where the eyes droop. And this is because the muscles that move the eyes are extremely energy requiring and very sensitive to any loss of capacity to make adenosine triphosphate. So I mentioned those in passing. More important from the point of view of survival of mankind is to understand and how to deal with neurodegenerative disease, Parkinson's disease, Alzheimer's, and Huntington's. Now, what is very clear is that the etiology, not the etiology, but associated with each of the diseases is something to do with mitochondria. We don't understand what yet. Without any doubt, one form of Parkinson's, so Parkinson's disease is not a single disease. There are familial forms, that is to say specific mutations which are inherited, are associated with Parkinson's disease. Those mutations affect the functioning of the mitochondria. And it is a mitochondrial defect that is associated with familial Parkinson's. But the same is not necessarily true for the more common sporadic Parkinson's. It's also clear that the proteins that accumulate in Alzheimer's disease, the A-beta peptides, they get into mitochondria. And people think or have proposed that they inhibit the ATP synthesizing machine. I'm not convinced about that. And likewise, in Huntington's disease, alpha-synuclein, which is the protein that accumulates, also gets into mitochondria. And people have proposed again that it inhibits the ATP synthesizing machine. We've looked at that, and we don't think it's correct. Now, there also is a much gathering pace is the involvement of mitochondria in cancer. This is becoming a major area of cancer research, and I'm going to speak about it in detail. And then for a long time, there's been the idea that the aging process is something to do with the breakdown of the DNA inside mitochondria. And people used to think that it's because it was being bombarded all the time with a form of oxygen that is generated inside mitochondria called reactive oxygen species. And it was thought that the reactive oxygen species bombarding the mitochondrial DNA caused it to mutate. We now know that that is not correct, although the idea is very widespread and people think that it is so, but quite clearly coming from work done in Stockholm and in Cologne, the mutations do occur, but they're actually occurring because of incorrect replication of the mitochondrial DNA, so that during life we're accumulating mistakes, somatic mutations in our mitochondrial DNA, which means that as we get older, our capacity to generate energy is reduced, and undoubtedly it is part of the aging process. Now let me link back to my Oxford days. So you heard that I was a student at Oxford, and you may be surprised that I did my PhD in the pathology department in Oxford. Well, the pathology department in Oxford was where penicillin was developed. So of course we're all familiar with Fleming's famous experiment in 1928, where he had grown Staphylococcus aureus on an agar plate, and a penicillium flew in through the window, landed on his plate, and he could see this zone of inhibition of Staphylococcus aureus, which led to the idea that there was an antibiotic preventing growth of Staphylococcus. What did Fleming do to turn that into a drug? Well, the answer is nothing. He actually did not pursue his own idea. So although he later took credit for developing penicillin in clinical medicine, and every school child thinks of Fleming when you mention penicillin, actually it was due to the efforts of two Oxford scientists called Howard Florey and Boris Chain. And in 1939, they selected 10 principles that had been described in the literature which had antibiotic properties, and they wanted to see whether they could develop them for use in clinical medicine. And one of those 10 was penicillin. One of the other 10 was what I did my PhD on. It got nowhere in clinical medicine, although it produced a PhD for me. Now, the penicillin was very quickly, under these difficult wartime conditions, the organism was grown and penicillin was isolated. And my PhD supervisor later, Edward Abraham, in 1940, before penicillin had ever been used in clinical medicine, showed that Staphylococcus aureus rapidly became resistant to the effects of penicillin. So penicillin resistance was detected almost as soon as penicillin was isolated. And it wasn't actually put into clinical use until 1943 or 1944 when it was used in the battlefields in North Africa. And then, of course, in 1945, Fleming, Chain, and Florey received the Nobel Prize for the discovery of penicillin and its curative effects in various infectious diseases. Now, this story of the emergence of resistance has continued. So as each antibiotic has been introduced into clinical medicine, so here it says penicillin 1943 and then 46, but I know that Abraham discovered resistance before this date as I've described. But if you go down this list and compare the dates here, almost as soon as an antibiotic is brought into clinical practice, resistance is detected very quickly. And the reason for that, of course, is what the antibiotics are doing is to kill off the sensitive organisms, but the resistant organisms are already there. They're not mutating in response to being exposed to the antibiotic. They're already there in the population. They've arisen by random mutation. And what you're doing is selecting the resistant microorganisms. So here's more information about how resistance has increased. So this is MRSA, multiple resistant Staphylococcus aureus, ramping up like this. VRE is vancomycin resistant Enterococcus, similar phenomenon. FQRP, fluoroquinoline resistant Pseudomonas, same story. Here now are the annual deaths arising from antimicrobial resistance in 2014, so fairly recent. Here we are, 700,000 people worldwide died from antimicrobial resistance. Not quite as many as die from car traffic accidents, more than die from measles or cholera. But look, if we do nothing, by 2050, it's now 10 million people a year dying from antimicrobial resistance, more than the total deaths worldwide from cancer today. So this is a serious problem. And here, the impact by 2050 will not be felt evenly throughout the world. The impact will be felt mainly in Asia and sub-Saharan Africa. Here are some more recent figures from the UK, from England. Number of antibiotic-resistant blood infections, 2013, 14, 15, 16, 17. Now, in 2016, I got an MRSA infection that very nearly killed me. And I was extremely ill from a very simple wound that became infected in the hospital by MRSA. So I know from first hand that it's not fun. Now, why is resistance spreading through the world? Well, there are a number of explanations. One is that clinicians have overprescribed antibiotics, often from pressure from patients. And then patients often don't finish their treatment. And then this, I think, is a major problem, is the overuse of antibiotics in farming. And then, of course, there have been other simple issues like poor infection control in hospitals and clinics. This has improved immensely in the UK in the last 5 years. And then another reason is there's actually a lack of new antibiotics being developed. So this shows you in this particular picture the difference in consumption of antibiotics in different European countries. So antibiotics prescribed in primary care, daily dosage per 1,000 people across this period of time. So the brown, I don't have Denmark, I'm afraid, but this is what is it? This is Greece and France here, so they consume considerably more antibiotics than in the United Kingdom, Germany, and and the Netherlands. So and then here is some information about misuse of antibiotics in the farming industry in the USA. Animals consume twice as many medically important antibiotics as humans. And these antibiotics, this 70% of the antibiotics that are being consumed, are not being used to cure animals, they're being used prophylactically. So they're just given to animals to keep them healthy and to make sure that they continue to produce milk and grow fat. And of course, what is happening in that process is that the resistant strains of microorganisms are being selected on an industrial scale. And of course, then the resistance genes transmit themselves horizontally to microorganisms that affect us. And then the issue about discovery. So whilst in the 1940s, 50s, up to the 60s, there was a flurry of discovery of all kinds of very important antibiotics. From this point onwards, people either became overconfident or the drug companies felt it wasn't worth their while, or a combination of the two. But very little happened between the mid-60s and the mid-1990s. There are very few antibiotics discovered in this period. The task has now been taken up again for obvious reasons. Now let me come to tuberculosis. Now, I can remember as a small boy people in my village dying from tuberculosis. One of my uncles actually died from tuberculosis. And then once streptomycin came along, it just disappeared. We've never heard of it in UK populations. It's a disease of the respiratory system, spread to the brain, kidneys and bones. It's highly contagious, chest pain, fatigue, weight loss. Now look here, 2 billion people are infected in the world today. And in 2017, 1.6 million people died from tuberculosis. And 60% of those who are untreated die. So currently, so this is from the British Medical Journal recently, 490,000 people now have multi-drug resistant tuberculosis. This is getting really serious. Almost no antibiotics are useful to these people. And in the last 3 years, there have been 20 cases of totally resistant tuberculosis arising in the Indian subcontinent. And I told you about the death rate in 2017. And I've also pointed out that specific parts of the world, China, India, and Russia, bear the brunt of this problem. That's emphasized in this picture, which says the same thing, really, that India, that Asia and sub-Saharan Africa bear the brunt of the problem. And the problems are multiple resistant tuberculosis, and then the association of tuberculosis with people who got HIV. They're immune compromised and much more susceptible to infections. Another problem with Mycobacterium tuberculosis, the causative agent, is that it has two states of life. It has an actively growing state when it's dividing in the body, but it goes into a cryptic state where the organisms, they're in green in these pictures, become surrounded, encapsulated by these other cells, by macrophages and neutrophils and T cells and B cells. And in this state, the Mycobacterium are not dividing. They're in a quiescent, dormant state. The problem then is that if you treat someone with a drug that is effective against tuberculosis, it will kill the living microorganisms, but these ones escape. And then at a later stage, they will reactivate, and the drug will re-emerge from somebody who thinks they're already cured. Now, the impact of dormancy in this problem has been questioned recently, actually, by the sister of Venki Ramakrishnan, who is an expert in tuberculosis. So it's not so clear that this is a major factor, but it is a factor. And so why is there a resurgence? There's poor adherence to TB control programs. Remember where these problems are occurring in the less developed parts of the world. Drug resistance, multi-drug resistance, extreme and total drug resistance. White explained this synergy with HIV. It's difficult to diagnose, and the drugs and vaccines are outmoded. So we need new drugs against new targets and new vaccines. So the vaccine that's in current use is BCG, which was developed at the Pasteur Institute in 1921. And we need better and earlier methods of diagnosis. So, I mean, apart from my background at Oxford with E.P. Abraham, why am I interested in this? Well, here we are. When was it? I think it was 2007. This paper appeared in Science, a diarylquinoline drug active on ATP synthase of Mycobacterium tuberculosis. So this drug is called bedaquiline, and it's a very effective anti-tubercular drug. And it shortens the treatment. So one of the current issues, many issues in treating tuberculosis, is that people have to take the current drugs for up to 6 months. In this case, the treatment can be shortened down to months or even weeks with this particular drug. So this drug, amazingly, inhibits the ATP synthesizing enzyme in the Mycobacterium inside a human without affecting the ATP synthase in your mitochondria. Now that's something that would never have been predicted. So it's, as I said, it's called bedaquiline. It was the first approved anti-TB drug for 40 years. And it's highly specific for Mycobacterium, active against both replicating and dormant states of Mycobacterium, and is active against extreme and multi-drug resistant Mycobacteria. That's its chemical structure. However, there's always a downside. Bedaquiline is not a wonder drug. It was approved by the FDA extremely rapidly on the basis only of phase 2 trials with 440 people, such is the dire need for a new drug. And it does cause death in about 10%. That death is unexplained. There's a cardiovascular risk. And it's only used in these extreme cases of extreme and total drug resistance. It's a very lipophilic molecule, and that means it persists in the human body for up to half a year, which would not be acceptable pharmacologically for most drugs. And patients become susceptible to side effects. And so the bottom line is that we need more inhibitors that inhibit the mycobacterial ATPase in different ways. So I've not said this, but what bedaquiline does is actually to bind to the rotor ring and prevents the turning of the rotor. Now the rotor in Mycobacterium is C9. It's got 9. We've got 8. So this drug is able to distinguish between a C9 ring and a C8 ring. So what we've started doing is to determine the structure of the rest of the enzyme. Here it is. This is the catalytic part of the ATP synthase from Mycobacterium smegmatis, which is a non-pathogenic Mycobacterium closely related. And it looks, for people who are familiar with looking at F1 ATPase, the catalytic part, it looks like every other one. But you need to look at the details, and there are subtle differences. Now, it turns out that people have studied bacterial ATP synthases very little relative to the amount of work that we and others have done on the mitochondrial enzyme. Bacteria live in very diverse conditions of pH, oxygen, and salt. Some are driven by proton motive force. Others that live in alkaline conditions make what we call a sodium-ion motive force. Now, importantly, some of them, like E. coli, they can both make and synthesize and hydrolyze ATP. The rotor will turn in both directions. But Mycobacterium is different. It will only synthesize ATP. It will not do the reverse reaction. Now we look at that structure. We're very familiar. Why doesn't this thing hydrolyze ATP? We don't have an explanation. Currently, we're working on that. In other words, the regulatory mechanisms in Mycobacterium and in other microorganisms. So this strategy could be applied to Staphylococcus aureus. We could look for drugs to treat Staphylococcus aureus based upon the ATP synthesizing complex. So but we need to understand what are the differences between the mycobacterial enzymes and the human enzyme so that we can exploit those differences. So that's one of my major interests at the moment. I'm running over time. I'll rush through the energy crisis. So the message is that the world population has risen enormously since AD 1. Here it is going up at a rate of 1 billion every 13 years. But it's even worse than that, because as nations become developed, so the United Nations have something they call the Human Development Index, which includes life expectancy, literacy, enrollment in schools, and GDP per capita. As this Human Development Index goes up, so does your need to consume energy. So the most energy-consuming nation in the world is Iceland, perhaps for obvious reasons. I think Denmark is in this cluster here with the UK and Germany. But as living conditions improve in China and India, they will rise relentlessly up this curve. And they'll need more energy to keep themselves going at the levels of that they require for their lifestyle. Now, currently 85% of global energy comes from fossil fuels. These are the distributions here, most of it coming from oil. These are our demands for worldwide for energy, 15 terawatts, which is equivalent to 150 billion 100-watt light bulbs burning continuously. And by 2050, our requirements will have doubled to 30 terawatts. Now look at this picture. This is really alarming. So this is the relentless rise of carbon dioxide in the atmosphere as measured in Hawaii. And this is 1965, this is 2015. These are the levels in the atmosphere. Now this sawtooth is explained here. In winter, when there's no photosynthesis going on, the levels get higher. And then in summer, when the trees and plants are green, they're fixing carbon dioxide and it goes down again. But the general trend is alarmingly upwards. So here are potential sources of carbon-free energy. Hydro, tides, wind, geothermal, we're exploiting all of them. Solar, and there is current use, and then the expected doubling of the use in the next 20 years. A lot of hope has been placed in atomic energy. So here's an atomic energy station in Virginia. What it generates is 1,500 megawatts. There are all kinds of problems associated with atomic power stations. We know waste disposal, security, and safety, as illustrated by Fukushima. But look at the top. We need to build one a week for 200 years to supply all our present energy requirements. And at present, it takes more than 10 years to build one atomic power station. So this doesn't look to me like strategically like a good long-term solution. It may be an interesting and useful interim solution. The answer is here. The sun is a nuclear fission reactor 93 million miles from Earth. 1 hour of sunlight equals our annual consumption of energy. The good news is that planet Earth is energy rich, and we need to get better at harvesting the energy that's falling on the Earth. Thank you very much.